What are cells and viruses? In biology, a cell is the smallest unit that can live on its own that makes up all living organism and the tissue of the body. Living things such as human, animals, plants and bacteria are made up of cells. A typical cell is a mass of cytoplasm that is bound externally by a cell membrane, and cytoplasm usually contains organelles such as mitochondria, Golgi apparatus, endoplasmic reticulum, ribosomes and many more. Genetic material can be found in enveloped nucleus for eukaryotic cells or in nucleoid region of cytoplasm for prokaryotic cells, which is critical in formation of an organism. Cells can be categorized into two main categories, eukaryotic cells and prokaryotic cells. Prokaryotic cells are smaller and simple in terms of structure, and examples are bacteria and cyanobacteria. Eukaryotic cells are larger and more complex. Animal cells, plant cells, fungi and protists are eukaryotic cells. Viruses are microscopic parasites, generally much smaller and simpler than cells. They lack the capacity to thrive and reproduce outside of a host body. Viruses are biological entities that can only thrive and multiply in a host, which is a living organism such as a human, an animal, a plant or even a bacteria. A typical virus consists of a core of genetic material, either DNA or RNA, which is covered with capsid, a protein protective coat. Envelopes may present a round capsid to help viruses to bind and infect cells. Viruses come in many shapes and sizes, but these are consistent and distinct for each viral family. In general, the shapes of viruses are classified into four groups: filamentous, isometric or icosahedral, enveloped, and head and tail. Filamentous viruses are long and cylindrical. Many plant viruses are filamentous, including TMV, tobacco mosaic virus. Isometric viruses have shapes that are roughly spherical, such as poliovirus or herpes viruses. Enveloped viruses have membranes surrounding capsids. Animal viruses, such as HIV, are frequently enveloped. Head and tail viruses infect bacteria such as bacteriophage T4.
Now, let us look at the relationship between cells and viruses. Viruses need to rely on cells in order to survive in the environment by living inside host cells. If viruses are not in host cells, viruses cannot replicate and thus cannot survive for long. This kind of relationship can be both beneficial and harmful. The first benefit of relationship between viruses and cells are viruses can be used to cure cancer. In the diagram shown, oncolytic virus is used to infect and destroy tumor cells without affecting healthy cells. The second benefit is that viruses can be used to correct genetic disorder in the cells. This is also known as gene therapy. For example, adenovirus vector can be used to deliver gene into cell nucleus and the genetic disorder can be cured. This process does not harm the cell as the virus is genetically engineered. Third benefit is that viruses can be used to treat bacterial infection via phage therapy. Bacteriophage virus is used to target and destroy bacteria in host cells without causing detrimental effect to the host. Let's look at harms of the relationship. The first harm is that viruses can infect and cause disease in cells, and eventually lead to cell death. The mechanism of infection will be discussed in more details later on. The next is if virus of a disease is spread extensively, this can cause global pandemic or public health emergency. The most notable example is COVID-19 pandemic that has been happening since December 2019. How can viruses be spread among cells? The first mode of transmission is direct transmission, which can be further split into two subcategories. The first subcategory is direct contact with people or living things that are infected with viruses, and this involves skin-to-skin contact, kissing and sexual intercourse. Another one is droplet spread when infected people or living things are sneezing, coughing or talking over a few feet distance. The second mode of transmission is known as indirect transmission. In indirect transmission, there are three subcategories that are usually well known. The first subcategory is airborne transmission, where viruses can be carried by dust or droplet nuclei suspended in the air. Next, viruses can be spread via food, water or biologic products such as blood via vehicle-borne transmission. After that, viruses can live in vectors such as mosquitoes and ticks, and this is known as vector-borne transmission. Now let us look at natural reservoirs or hosts of viruses. Animals such as bats and primates such as monkey are known to be natural reservoirs of various types of viruses. Based on the research, it was found that those animals have more permissive immune system that allow them to carry high load of viruses which can be potential threats to humans. If a virus carried by them can be transmitted to humans, the virus is known as zoonotic virus, which can cause zoonotic diseases. In fact, patient zero of most diseases can be traced back to natural hosts or reservoirs. The most notable example of natural reservoirs is bats, which are responsible for many viral diseases such as Ebola and SARS coronavirus 1. Ebola can cause hemorrhagic fever, whereas SARS coronavirus 1 is responsible for severe acute respiratory syndrome disease. Bats are also suspected to be the culprit of current COVID-19 pandemic outbreak. Why are bats known to be responsible for most diseases? It is because they can fly, have unusually long lifespan, are consumed as bush meat in some regions such as China and Africa, and their distribution is widespread in nature. Monkeys are also known to be responsible for Zika virus outbreak in America continents, and person infected with Zika virus may suffer Zika fever. Dogs can be responsible for rabies disease if they are infected with rabies virus and transmit disease to humans. The following tables show more examples of natural reservoirs and viruses, which are taken from the main review article of the video. Please kindly pause the video if you want to look at the table in details. How do viruses infect cells? In general, there are 6 main steps that we need to take note. First, a virus attaches to the cell surface, penetrates the cell plasma membrane and enters the cytoplasm. Next, envelope of the virus is stripped off by substances in the cell. This is then followed by the release of viral genetic materials into the cell cytoplasm, and the viral genetic materials can be either DNA or RNA. In order to cause disease in cells, viral genetic materials must enter the cell nucleus. This subsequently causes the cell to ignore its actual cellular function and start to make more viruses. Then, the infection reaches a certain extent that the virus is released from the cell and the cell is destroyed in the process.
Now, let's take a look at how cells defend against viruses in human. Natural killer cells are created in the human body as part of the innate immune system. The process involves a collection of receptors located on both healthy and diseased cells, as well as natural killer cells. During the early antiviral response, natural killer cells are important cells that produce effector cytokines and eliminate infected cells. Natural killer cells is a type of white blood cell that has granules with enzymes that can mediate resistance against intracellular pathogens, mainly viral infected, bacteria infected and protozoa infected cells, and have the potential to restrain cancer and metastasis. Here we can see the receptors found on natural killer and healthy or infected cells. Natural killer cells possess germline-encoded receptors that can be activating or inhibitory. Healthy cells possess few activation ligands and more major histocompatibility complex class I molecules MHC1, while cells infected by viruses have no MHC1 or MHC1 fewer than activation ligands. As we now understand the acting receptors of natural killer, healthy and infected cells, it is time to go through how natural killer cells work as a part of the immune system in human body. In order to recognize healthy or normal cells, activating receptor of a natural killer cell binds to activation ligand of a cell, and inhibitory receptor of a natural killer cell binds to the MHC1 of the cell. Because more MHC1 attach to the inhibitory receptors of natural killer cells and exerts a strong inhibitory signal on them, the cell with more MHC1 is detected to be healthy and will not be killed by natural killer cells. MHC1 is absent or present in lower amounts when a cell is infected by viruses, which can be categorized as reduced inhibition, and more activation ligand binds to the activating receptor of natural killer cells. As a result, a type of peptide called cytokines activate natural killer cells, causing them to generate granzymes and perforin, which kill virus-infected cells by lysis. In strong activation, infected cells can sometimes boost the expression of molecules recognized by natural killer cells activating ligands, allowing them to receive a much stronger activation signal than usual. This could cause the natural killer cells to act by overriding the inhibitory signal. Natural killer cells also emit cytokine IFN gamma to warn surrounding tissue of infection.
We can actually vaccinate against viruses by utilizing natural killer cell compartments. For instance, a zoonotic virus is an infectious virus that can be transmitted between humans and animals. Among the biggest challenges we face with zoonotic viruses is we are lacking of effective vaccines. Until today, we only have effective viral vaccines against 5 diseases, which are rabies, tick-borne encephalitis, Japanese encephalitis, yellow fever, and Ebola viruses. In prime boost studies where 3 doses are recommended for full protection, natural killer cells are among the first and strongest responders during each revaccination dose. Interferon gamma cytokine production and degranulation predominantly occurred. A study on an effective vaccine against Ebola developed in Canada which was approved for use in the US and Europe shows there is a correlation between a strong natural killer cell activation and development of an antibody response to this Ebola vaccine. Another study on 2 recombinant rabies-based vaccines which are being developed against Marburg and Lassa viruses shows that antibodies produced in the body stimulates strong natural killer cell-mediated response against infected cells possessing the viral glycoprotein used for vaccination. These studies highlight the critical role of natural killer cells in conferring protection through vaccination against zoonotic diseases.
In conclusion, cells and viruses are an inseparable part in nature, and their relationship can be described as a double-edged sword. Thus, relationship between cells and beneficial viruses can be utilized for our benefits. At the same time, more research should be done to develop more effective therapies or vaccines against bad viruses, especially bad viruses that have not had effective therapies or vaccines yet. That's all from us and thanks for watching.